we are about ready to start. I'm Paul Beck, and I'm the chair of the steering committee for the Emeritus Academy, and I have the pleasure of introducing our speaker today. But before I do that, just a couple of things. One is that this is the second talk in our Emeritus Academy Lecture series, and we invite you to come every month, first Wednesday of the month, to these various talks, and on our website and in other places you will see information on these talks. A wide variety of topics drawing upon the incredible expertise that resides within the Emeritus Academy. I would invite you to help yourself to food in the back, but I think you've actually done that. Just leave some Buckeyes for us afterwards so that we can retire to that. They're gone, okay. And I also invite you to stay after the talk for a very, very short business meeting. I promise that it will be, 2 or 3 minutes, and probably no more than that. Our speaker today is Gary Bernsen from the Department of Psychology, a member of the Emeritus Academy. He retired in 2010, but really didn't retire, as is the case, I think, of people in this room. He's a neuroscientist. His more recent work focuses on anxiety and autonomic control in both humans and animals, I think. Like many others in this room, as I said before, he's continued to be an active scholar and researcher. The 4th edition of his co-edited Handbook of Psychophysiology was published by Cambridge University Press in 2016, and I invite you all to go to Amazon or whatever and buy it, of course. And he has a variety of refereed articles and book chapters. One thing of special note that I wanted to mention here is that he's one of these scholars and researchers whose research has served the public interest by talking to policymakers and others about the things that he has discovered in his own research program. He is a member of the National Academy of Sciences Committee on Social and Behavioral Sciences and National Security. He was on the Defense Department's Committee in Predicting Violent Behavior in the aftermath of the Fort Hood shooting, something of real relevance, I think, today, continuing relevance. He's been a member of the Department of Homeland Security's Attribute Screening Technology Program. Today, what Gary is talking about is non-invasive brain stimulation, including its use in performance enhancement in athletics, and the issues that arise from it. He notes the potential applications extend well beyond this particular topic. Speaking personally, I will be listening for guidance as to how I can improve my competitive edge, as I think many of you probably will be. Well, without further ado, Gary.
Thank you for that generous introduction. I want to talk brain stimulation today. This is a talk that grew out of an invited presentation to the U.S. Anti-Doping Agency, who are the folks that took down Lance Armstrong, and Sports Medicine Research and Testing Lab. These are the folks that do the drug testing for the NFL and other professional teams. If it isn't apparent now why they might be interested in this, it will become so shortly. A bit of history. Brain stimulation has been around for over a century. We had some early applications in the 19th century. But most of the significant systematic work has emerged from the early 1920s. Notable among which is work of Wilder Penfield, who was a Canadian neurosurgeon. He was actually born in the United States, but he moved to Canada and spent most of his time at the Montreal Neurological Institute there, where he was studying surgical interventions for epilepsies. And in the course of that enterprise, he would stimulate local areas on the cerebral cortex exposed for certain neurosurgical procedures in conscious patients who had just a local anesthesia. The purpose was to identify critical functional areas that would be spared in any surgical procedure, or to identify epileptic-like loci. It was his work that gave us this legacy of the well-known sensory and motor homunculus that you see in every introductory psychology and biology textbook. A couple of other early developments. The work of Walter Hess, who developed methods for chronically implanting depth electrodes in the brain. He received a Nobel Prize in Physiology and Medicine in '49 for his work that identified systems that regulate not only visceral autonomic function, but affect, emotion, including limbic area circuits like the amygdala to the hypothalamus to the midbrain central gria, quotes, rage circuit. Come back to that. This work has led to contemporary deep brain stimulation procedures, which we'll visit in a moment. Hess shared that '49 Nobel Prize with Igaz Muniz, one of the early developers of psychosurgery, which was believed at that time at least to hold promise in the treatment of psychological disorders. Fortunately, this brutal approach fell into disfavor in the late 50s and early 60s for a number of reasons. First of all, behavioral circuits that underlie complex psychological processes aren't punctate. There isn't a place you can go and find a manipulation that's specific to that. But out of the ashes of psychosurgery, and in conjunction with brain stimulation, arose the phoenix of functional neurosurgery. Psychosurgery is a surgical procedure designed primarily to alter psychological or behavioral processes. It's not a viable strategy. Functional neurosurgery is neurosurgery designed to rectify, correct, or mollify a neurological abnormality. If that changes behavior or psychology to the good, all the much better. But the primary purpose is to deal with that physical, neurological condition. Who remembers Charles Joseph Whitman, the Texas sniper? We've been reminded tragically of him again recently. In some cases, the contemporary press would have us believe that violence, especially school violence or mass murders, are a recent development arising out of our political economic milieu. Not true. 1966. Whitman climbed the tower at the University of Texas at Austin, killed 17 people, 2 died after this publication of this newspaper, wounded 31 others. Who can tell me the worst school massacre in the history of the United States? Nope. The Bath School disaster. Andrew Kehoe, who with a knife and bombs and fire killed 44 people, most of them students, and injured 58 more. That occurred in May 1927. The worst school massacre in the history of the United States. So at autopsy, the coroner noted what he claimed was an astrocytic tumor. But he says this wouldn't have had anything to do with Whitman's behavior. But then Governor Connolly, recall the survivor from a bullet wound occurring during the Kennedy assassination a couple of years earlier, commissioned a panel of experts who reevaluated the evidence. First of all, it wasn't an astrocytoma, it was a glioblastoma multiforme, and it was located in the vicinity of the hypothalamus, impinging amygdala, temporal lobe, some of those areas that Hess had identified in his rage circuit. Did this have anything to do with his behavior? We'll never really know. But the report does concede the tumor could have contributed to his inability to control his emotions and actions. It could have enhanced impulsivity. So from Whitman and other similar cases, we see one functional neurosurgical approach illustrated by Vernon Mark and Frank Irvin. Mark, a neurosurgeon, Irvin, a psychiatrist at Harvard Medical School, who were interested in the potential association between violence and brain dysfunction. They had a series of patients. One notable is Julie, a 21-year-old, back in the 70s. Had a history of epilepsy since an encephalitis attack at age 2. An epilepsy then sometimes referred to as psychomotor epilepsy, now more commonly referred to as partial complex seizures, partial or simple or complex seizures, which may involve momentary lapses of consciousness, stereotype repetitive behaviors, or even complex acts occurring during the epileptic episode or during the post-ictal fugue-like state following the end of the epileptic episode. Mark and Irvin noted abnormal electroencephalographic activity around the amygdala, both sides of the brain. Implanted depth electrodes stereotaxically into the vicinity of the amygdala where these abnormal activities seem to be arising. Electrodes through which they could both record and stimulate and, in fact, burn neural tissue. This is Julie, amygdala, EEG, when she was calm and at rest, with stimulation of one of the regions around the amygdala illustrated by the stimulus artifact here, the dark wide lines. Julie's demeanor changed. A seizure was initiated and a violent outburst ensued. Based on these results, Mark did a focal ablation around the tip of the electrodes, both ended up bilaterally, where these epileptiform-like activities could be evoked, which resulted in a dramatic reduction in seizures and aggression, at least for the 2-year follow-up that they had with her. Functional neurosurgery, an effort to correct a neurological abnormality, which secondarily had benefit, behavioral benefit. Julie had taken to carrying a knife because of these fugue states. She's end up in unknown locations, and she got referred because of multiple stabbings, largely resolved following the surgery. So, what is depth stimulation like today? It's widely used for a number of conditions. Non-Parkinson essential tremor, Parkinson's disease, approved by the FDA, 1997, 2002, dystonia, treatment-resistant epilepsy, psychiatric disorders, chronic pain, although that's not yet approved by FDA, there are a number of studies of positive effects of deep brain stimulation. We have a neuromodulation group here. Unfortunately, the director is heading to West Virginia to join our friend out there, but we still have a solid group doing deep brain stimulation. Let me give you one example of a current deep brain stimulation method referred to as a NeuroPace RNS system. It was developed by folks at Stanford and who then established this NeuroPace tech group. This is a device, the RNS system, device where electrodes are implanted based upon surface EEG recordings that suggest foci for epileptogenic-like activity. Electrodes are implanted. Those electrodes can both record and stimulate, much like Mark's. There's a monitor device, a computer-controlled monitor that's recording EEG. If it detects abnormal EEG patterns, which can be adjusted or set for an individual patient, it initiates stimulation to disrupt that ongoing pattern. And that stimulation can be adjusted to achieve a diminution of the abnormal electrical activity. The device can be charged and data read inductively through the scalp. They report average seizure control of about 60% over a 3-year period. One additional perspective. Deep brain stimulation or even surface stimulation involves a direct activation of neural structures. But we know that there is a massive set of ascending afferent pathways some of them running by the afferent vagus nerve, but multiple other pathways as well, that send ascending projections to higher level systems known to impact cognition, emotion, and general cortical excitability. The details here are not important, just want to illustrate the profound influence that these ascending projections can have. Do we need to go inside the scalp, is the question. And we now have a system that can drive central networks used for intractable epilepsy, an electrode around the peripheral vagus nerve, a so-called VNS implant subcutaneously, sort of like a vagal nerve pacemaker. Approved by the FDA for epilepsy 1997, for drug-resistant depression in 2005. Considerable studies emerging on potential benefit in heart failure and pain syndromes. Here is a case where we can drive central networks without an intracranial electrode. As a matter of fact, you don't necessarily even need the vagal electrode. This can be accomplished by the gamma core inductive system by simply placing this inductor up against the neck. And that has been shown to have potential effects in treatment of migraines and cluster headaches. Or we can stimulate that vagus nerve by the auricular branch of the vagus nerve, which innervates the concha of the ear. Induces very characteristic activity in a brain structure called the insula. And initial results indicate positive effects on depression. We'll see where that goes. But the general strategy here is one that Defense Advanced Research Projects Agency is now looking at very closely. What benefit might there be to activating these afferents, particularly in conjunction they're interested in with neuro modulators? Can we improve synaptic plasticity? Can we improve recovery from battle injuries, for example, or enhance performance? So this is a non-invasive brain stimulator, the Fisher-Wallace stimulator. It's a Canadian outfit. Don't put it on your head. It's been FDA cleared, they claim, as a Class 2 medical device. That doesn't mean FDA approved for any specific purpose, but it has been cleared, and there is a growing literature on potential applications of non-invasive brain stimulation to treatment. Or, if you don't want to go to Canada, go to England or have them ship over one of these, a far, far more sophisticated neurostimulator than the Fisher-Wallace device, based on an ARM microprocessor. You can control it by an app on your smartphone, or for that matter, Google Watch. It can produce a whole variety of stimulus patterns, so-called transcutaneous direct current stimulus, transcutaneous alternating current stimulus, and so on. Far more versatile than Fisher-Wallace. Well, what about FDA? They don't care about FDA. They're not marketing this as a medical device. They're marketing it to gamers to improve their video game performance. They're marketing it to athletes. An increasing literature now documents performance enhancements. They're small, but consistent, reasonably well-controlled studies. 4% increase in peak power output. This is in a bicyclist. 4% doesn't seem much, but it could mean number 1 versus number 2, or for that matter, number 10. Some of this associated with a diminished perception of fatigue, but in other cases you see performance effects independent of that perceived fatigue. Very broad, non-specific stimulation that will include the motor cortex and wider areas of the lateral cortex. Another one's come along. Chow and Wingier. They were with NeuroPace. They were engineers, helped develop that NeuroPace system. They see potential outside NeuroPace. They left NeuroPace, formed Halo Neuroscience, and sell a non-invasive brain stimulation device built into a headphone. Their motto is champions are built, not born. They partnered with the U.S. Ski and Snowboard Association. 20 minutes daily stimulation applied. 10 days. 13% increase in propulsive force. 11% decrease in wobble. Performance enhancing effects? Golden State Warriors think so. They are now exploring the potential performance benefit of the Halo neuroscience device. And I probably don't need to remind you that they just beat the Cincinnati Cavs for the NBA title. Could be just a coincidence, right? But the San Francisco Giants don't believe so, because they've now acquired a set of these Halo devices. Or you can look at the Defense Department, their so-called DIUX, Defense Innovative Unit Experimental, who's interested in testing non-invasive brain stimulation for performance enhancement. Ash Carter first described this initiative last July in 2016. And shortly thereafter, they contracted with this company named Halo Neuroscience to test whether non-invasive brain stimulation could enhance performance in combat warriors. In February of this year, a major push by the Navy, and we have We have SEAL Team 6, among other units, testing out performance effects of non-invasive brain stimulation. The Air Force has partnered with Rio Grande Neurosciences on transcranial stimulation research. We have a local representative here, Andy McKinley's with the Air Force, has a cross appointment with Ohio State. He's out housed at Wright Pat. They're looking at a number of aspects of airman performance. Particularly one of the notable recent findings was a study of multitasking, where individuals have to engage in activities with divided attention and adjust depending upon what's happening in other aspects of the field, and they find significant 30 to 40% improvement in multitasking performance with frontal brain stimulation. Generally, they're looking at broad performance, cognitive performance enhancement. Specifically, the Air Force is very interested in attention, in identifying targets. And they find that stimulation of the left frontal eye field enhances target detection, accuracy, and it dramatically reduces fatigue over time. There is a voluminous evolving literature on aspects of non-invasive stimulation, from the biophysics and biochemistry and biology up to performance. We are seeing an increase in worldwide attention to non-invasive stimulation approaches. Just back from the Society for Psychophysiological Research last fall, we had 3 major symposia on non-invasive brain stimulation. Best practices, neuromodulation, improvement in transcutaneous vagal nerve stimulation. There are now numerous clinical trials, cerebral palsy, aphasia, major depressive disorder, through safety, moving into efficacy trials. We have a growing literature on improvement of motor skill by transcutaneous electrical stimulation, a dramatic increase in the number of reports of positive benefit in post-stroke recovery of motor X. The Michael J. Fox Foundation is supporting investigative research into potential applications in Parkinson's disease. There are some initial preliminary findings. Reports are showing advantage in gait training, especially dual task, where there is an effort to improve gait while engaged in other activities like pulling a set of car keys out of your pocket or counting. And a growing number that suggests some potential benefit within the cognitive and affective components of Parkinson's disease, both in humans and animal models. Here we see right-sided TDCS in an animal model of Parkinson's disease, an enhancement in so-called CFOS staining, a general nonspecific marker of neural activation, increase on the side of stimulation, an improvement in tremor frequency, an improvement in locomotor ability. Sounds fanciful, doesn't it? How can something as nonspecific be doing all of these supposedly wonderful things? Well, the emerging perspective is it's precisely because of that non-specificity that you can see this myriad of outcomes. There was an argument raised several years ago. How am I doing for time? Okay. Where Budzaki implanted electrodes into the brain of a cadaver. And then he applied transcutaneous stimulation. And he says, very little of it gets in there. This is true. But Budzaki, you didn't have to waste a perfectly good cadaver. We've known this from decades of stimulation during neurosurgical procedures. You got about a 10-fold difference between sensitivity to direct cortical stimulation and transcranial stimulation. What this non-specific stimulation is doing is changing the sensitivity of neural systems. It's not initiating action potentials. It's not directly driving neurons. It's modulating the sensitivity so that ongoing behavioral brain states can more efficiently occur. That's the sort of prevailing emerging perspective on how one can have such a diverse set of outcomes. You don't see performance benefits in sports without associating this stimulation with practice. Actually, you usually precede the practice with the stimulation, although sometimes people do it simultaneously. So this is an interaction between a physical stimulus and ongoing brain state. Here's a busy one. You want to spend the next hour talking about this? I won't. Paul won't let me. But just very briefly, what do we have going on here? We don't know. There's all kinds of things going on with neurotransmitters, with multiple systems. But we do have one phenomena in neuroscience that has served as an important model for neural learning, and that's so-called long-term potentiation, where a priming stimulus can result in an evoked response that is larger than before the priming stimulus, and that persists for some time. And we have decades of research. We know a lot of the players, brain-derived neurotrophic factor and its receptor, AMPA, and NMDA receptors. And all of those same players are mobilized with non-invasive brain stimulation. We got a long way to go to understand precisely what's happening, but we've got some interesting neural models to begin with. So I'll end where I started. Is it doping? Thanks for your attention.
We do have time for questions. Gary, do you want to call on people? And please state your question clearly.
Do you have any idea what the nature of the sonic attacks that have occurred in Havana, Cuba might be?
No, I don't. And actually, I've got a slide that should have shown up, but it didn't. One of the emerging methods in non-invasive stimulation is focused ultrasound, where a ultrasound generator can generate a sonication pattern that's focused and concentrated at a local position, which allows the physical activation of deep brain structures without activating more peripheral structures. So there are a number of possible ways of impacting neural function without necessarily auditory signals or cues. But no, I have no idea whatsoever. Yeah.
My question is, you described enhancement of virtually every physical, mental activity with non-specific brain stimulation. There must be a downside. Are there suppression of things that we'd like to keep around?
Almost surely, but they don't get published.
Okay. Bingo.
Let me give you for example. If you have a manipulation that's going to improve vigilance and focused attention, we have limited attentional resources. It's certainly going to have a cost on divided attention. So, yeah, almost surely there are going to be circumstances and conditions where this is not going to optimally benefit. Yeah, Gary.
Of it going the other way, making it suboptimal.
Even, oh, absolutely.
You know, is such that relative to Rolf's question, would this not be a way to do cyber warfare, turning us all into Trumps? Idiots.
Gary.
Yeah, Gary, first of all, your office is still waiting for you. If you want to come back.
Pardon?
Your office is still waiting for you. If you want to come back. But anyway, can you give us a sense, since you read the articles, can you give us a sense?
Will you let me keep my office?
Yeah, we
John
did.
did.
Can you give us a sense of what the controls in each of these studies were?
Yeah, good question. Good question. Some of them, the controls are, some of the studies are much better than others. I tried to select ones that are reasonably well controlled. Here's a couple of examples. For the auricular vagal stimulation, the auricular branch innervates the inner part of the ear, not the outer part of the ear. So a control condition is some subjects with stimulation in the inner, some on stimulation in the outer, unless those subjects know the anatomy. They're going to feel exactly the same thing. And basically what you feel is a light tingling. Usually that dissipates over 10 or 15 seconds. So another control procedure is to deliver the same stimulus, but just for a very short period of time. So the subject feels that tingling, but then that feeling tends to dissipate anyway. Another approach to control is location. Even though it looks like these are very widely distributed, you do see some differences. For example, the vigilance study of McKinney, the left frontal eye field was really far more effective than other areas. So that's another way people have tried to control. Yeah.
Are the stimulations time dependent? You haven't said anything about time dependency in that if I apply the stimulation, can I then go out on the football
Yeah, great question. Very, very powerful time dependencies here. The question is, what about time dependencies here? The usual common practice now is 20 minutes before you start the practice, or a regimen of daily 20-minute. If you look at neural excitability, which people have done, a 5 to 10 second epoch of stimulation can alter sensitivity for very short period of time thereafter. It's really quite stimulus bound. But once you get out to 10 minutes or 20 minutes, now you can see a persistence of, for example, motor sensitivity that will well exceed the duration of the stimulus, maybe out to an hour or two. And then the concept is that even though that's going to probably dissipate, if you can get the training in, this is a philosophy anyway, the strategy. If you can get the training in, once you've got that motor skill training, the effect of the stimulus might go away, but the effect of that training may be preserved.
That's what learning.
Yeah.
Hey, Gary, Jack Wood, long time. no
Hi,
see.
has been a while, hasn't it?
Absolutely. I struck that vagal nerve, only about 10% of the fibers carry information to the brain. So it makes you wonder how that actually works. But the question then for me is there a placebo effect? And do you usually measure a placebo effect, as we would, for example, in a drug study?
Yes, but you misspoke. The vagus is about 85% afferent in humans, even higher in animals. So it is a massive afferent, minimal efferent. Placebo, yes, there have been some placebo control. They're a little bit trickier because you can feel this. So again, you use something like a locus that will not drive the vagus. You'll get the same perceptual phenomena. That's sort of how they've dealt with the placebo effects, and there are some. Yeah.
Can you say anything about the mechanism of the stimulation? I mean, you mentioned ultrasound, which involves physical motion, but is this electromagnetic radiation? Radio waves, microwaves, et cetera?
There are a couple of ways of imposing a stimulus on the cortex. One of them is magnetic. I didn't talk much about that. That's been around for a long time, where you can generate a magnetic pulse that will induce current flows in motor cortex and generate muscle twitches and things of the sort. Most of this literature is just DC current, low amplitude, 1 to 2 milliamps. Most of it is going to be shunted through the scalp. Very little of it is going to get in, but it is a direct current. The typical mantra is anodal stimulation is more effective than cathodal stimulation. But we know from neurophysiology, that's absolutely not the case. Anodal stimulation is hyperpolarizing. Cathodal stimulation is excitatory. But what you've got is, with this external placement, you're really delivering the anodal current to the proximal dendrites. And the axons of pyramidal cells are down where you see what has been called a virtual cathode. So you're really activating those pyramidal cells down at around the axon hillock. You're inhibiting them up at the dendrites.
If I could follow
Yeah.
up, if
Oh, because folks are starting to look at other patterns of stimulation. Alternating current, for example, where you're going to see a lot less capacitive polarization develop. Pulsatile, you can even do pulse pairs. That's where some of the, I think, more exciting stuff these days is how might we optimize these parameters? Yeah.
As a humanist, I'd like to ask an ethical question, so in a very different area. I was struck by your mention of Department of Defense as well as athletic uses, and I'm wondering, has any concern been raised that deep brain stimulation can lower usual boundaries of caution and therefore make subjects more vulnerable to injury and harm?
It's certainly conceivable, certainly conceivable. I think most of the thrust has been people want to get funded, people want to sell their devices. You know, you're going to be looking for improvements. I think as the area matures, we'll see more systematic examination of what could be, because one effect in one context could be positive, could be very different and maladaptive in another context. It's a good point. Yeah.
Gary, a lot of companies are now jumping on this bandwagon of using non-invasive recording methods to, quote, track your mood. So like electrical skin conductivity. And so they're producing devices that you can buy. Is there any evidence that they're getting together with these folks? So you're feeling a little sad, at least your device tells you you are, so then you use your other device to give yourself some brain stimulation?
Yeah, the media lab at MIT is looking at some of that. They've of integrating the two, and of course there's a dramatic proliferation of biological monitors these days.
Yeah.
That, as a matter of fact, the Robert Wood Johnson Foundation is funding a project that I'm on entitled The Essential Self, in which there is a great interest in not just monitoring or quantifying. That's a group quite apart, but how you would use that to improve. Yeah.
Has anybody looked at any studies at whether there's an increase in blood flow in the brain?
You know, I'm sure there would be, but I'm not aware of any.
Yes,
I'm-
there absolutely is. There absolutely is an increase in blood flow.
Oh,
There are studies out there. that-
okay.
Yeah. The Excitability in particular.
And how would you characterize those decreases? How are they distributed? How are,
Increase in blood flow.
pardon?
It's diffuse, just like the current density.
Oh, okay. Okay, thanks. Yeah.
In terms of your last question, how much are, for example, sports enforcement agencies investigating performance enhancement, non-invasive devices?
Well, that's why they had me there. They really don't know what to do with it. They really, really don't know what to do with it. They really put on blinders. They wanted to know, and then when I laid some of this stuff out, well, whoa, you know, is that really doping? You know, how is that different than exercise? And so it's so no, they're thinking about it. Some of them are, but they're not going very far with it.
Do you want to tell us how you would answer that question?
I don't know if it makes any difference whether you'd say yes or no. You're modulating the brain. You're changing something. Doping is really a concept that makes more sense from a sports perspective, not necessarily, you know, a sort of legal aspect of sporting and competition. You know, not so much a scientific issue. Bob?
Has this technique gone head-to-head against acupuncture or acupressure?
You don't see you don't see an awful lot of overlap. Nobody is, some acupuncturists are using electrical stimulation at acupuncture points. Fisher Wallace up in Canada will have an acupuncturist write you a prescription for one of their brain stimulators if you want one. But you're not seeing a lot of that in this literature. This is coming really more out of the neurophysiological, the biophysics actually, end of things than it is the sort of allied med or
I think we have time for one more question, but I would hope Gary would stay after if he'd want to come up and ask him other questions.
Oh, nothing, very simple question, a kind of a note. In relation to the pharmacological screening of the anticonvulsants, corneal electrodes were used in animals to test anticonvulsant drugs. Almost all current anticonvulsant drugs are tested by simple electrical stimulation and how much current is needed to block out the excitability. That's one comment. And secondly, where did you get these gadgets and how much does it cost?
say again?
Where did you get these gadgets?
Oh, you can get them. Fisher Wallace, because there's marketing it as a medical device, you do need a prescription. As I said, they'll find an acupuncturist who'll write one for you. That runs, I think, about $500–600. The British Focus is closer to $300, but they even have a more abbreviated one that you stick on the end of a 9-volt battery with a little microcontroller adjustment that you can get for about $100 or $200.
It's not that I want to go and buy one.
Halo Neuroscience is still trying to hold out. Their thing goes for about $1,000.
What I want to know is who pocketed the devices that were passed. And who is going to send them to the cabs to try to improve their performance. Well, I hope you will join me in thanking Gary for a really fascinating